So, so good morning, good afternoon, and good evening to all of the attendees. It's my pleasure to welcome you all in this workshop. The title of this workshop is Machine Learning Aided Advances in Synthetic Biology. So, as we all know, synthetic biology aims to understand, refine, control, re-engineer, and evolve nature. And data is a key element in this process. And in this workshop, we will be looking at how machine learning is making use of novel kinds of data and is enabling novel processing of those data for synthetic biology applications. The first of these talks pertains to a problem in bioinformatics, namely detection and prediction of COVID-19 using tapestry pooling. This talk would be given by Sabyasachi Ghosh. Sabyasachi is from IIT Bombay. He is currently a PhD student specializing in computer science and machine learning. Sabyasachi, I request you to please start your talk.
All right. I hope you guys can hear me. I'll just share my screen as well. Okay. So, are you guys able to see my screen?
Yes.
All right. So I'll just jump right in, guys. So yeah, we'll talk about Tapestry, which is a pool testing method for COVID-19. And yeah. So we did most of this work last year, and it was published recently in the IEEE Open Journal of Signal Processing. So what's the need for pool testing? So first of all, we want to expand the testing capacity, especially last year. This was really important so that we can identify and isolate as many people as we can and prevent the spread of the disease. And also pool testing reduces the test cost. And now, in the post-vaccine world, it's still important because, I mean, most of us are still working from home, and periodic screening of people in the offices or campuses, that will be very useful for preventing further spread of COVID. And pool testing makes it more viable. So, I mean, initially you have the individual testing, right? You have 6 people and you have 6 tests, and you get who is positive or negative, right? So can we improve on this? Can we somehow use lower number of tests? So there is this testing mechanism called Dorfman testing. So in Dorfman testing, you divide these n people into pools or groups, and you test these pools. So here the pool size is 2, and you have 3 pools. So once you get the results of the pool, you can see that if somebody was part of a negative test, then that person or that sample must be negative, right? But if somebody was part of a positive test, then we don't know which sample was positive, or maybe all of them are positive or only some of them are. So we need to retest these samples. So so here we see that we took a total of only 5 tests for 6 samples. So we saved 1 test. And in general, if there are n samples and k infected samples, then this mechanism takes order of square root of n times square root of k test in the worst case. And we take advantage of the fact that the number of infected samples is a lot lower than the number of samples to be tested. And this is an adaptive method, which means that the second stage test depended on the results of the test in the first stage. So you couldn't determine beforehand all of your tests. So that's why it is an adaptive method. So what are the benefits? So you save on the cost and you increase the testing throughput. However, because our test, which is the RT-PCR test, that itself takes 3 or 4 hours. If you do a two-stage test, then it will take 7 or 8 hours, which is somewhat not desirable. So can we improve on that? And the answer is yes. So we can have a single stage testing mechanism, or also known as non-adaptive group testing. So here you decide all the tests beforehand. So in this example, we have 4 pool tests for 6 people. And then you get some results that some of the pools are negative and some of the pools are positive. So these results need to be decoded in order to get who amongst these original samples were positive. So decoding mechanism is very simple. So you just look at who took part in a negative test. So if somebody took part in a negative test, then that person must be negative. And the remaining samples, we declare them as positive. So because all of their tests were positive. So this mechanism is called COMP, or combinatorial orthogonal matching pursuit. And this our pooling scheme can be represented as a matrix, which is a 4x6 binary pooling matrix, uh, and where a 1 just indicates that this sample took part in the corresponding test. And this matrix must have the disjunctness property for COMP to work. So what is disjunctness? So disjunctness means that for a k disjunct matrix, the support of any column is not a subset of the union of supports of k other columns. So so for example, this matrix is one disjunct. So if you look at column 1, then the column 1 is not subsumed by any other column. So if you compare it with column 2, then there is still this extra 1 here, which is not there in column 2. However, this is not a two disjunct matrix, because let's say I take the union of columns 2 and 4, then I can see that column 1 got subsumed by the union of column 2 and 4. So this matrix can decode up to 1 positive exactly, but it cannot decode more than that exactly. There will be some false positives. So, okay. So what are the benefits of COMP? So a major benefit is that it's one stage, and asymptotically it takes fewer number of tests than Dorfman testing, and that's a probabilistic guarantee. So how can we improve this? What more do we want? So it will be really nice if we also had the viral loads of the positive samples. So basically, these viral loads may be important for determining how severe the disease is in a patient, or for identifying which patients may be super spreaders. So, and in individual RT-PCR test, you can do that because they do give you a quantitative readout. But so we want that for pool testing as well. So for that, we need to go to compressed sensing. So what is compressed sensing? So in compressed sensing, initially your viral load vector of all the samples, that is unknown, right? And again, you do the pooling. Here we have the same pooling matrix as before, and you run your tests, and you get the viral loads in each of these pools. So now once you have viral loads in these pools, you would want to decode this and get back this unknown X star. So basically here this system is an underdetermined system. So in the previous example, there were only 4 equations and 6 variables. However, if my X star is sparse, so that means that most of its entries are 0, only k entries are non-zero. then I can do the decoding. But there is a further condition that my matrix A needs to be special. So A needs to have the property that no 2k sparse vector lies in its null space. And you can see that because of this property, it will have only a unique solution, because this A times x1 minus x2, if x1 and x2 were two solutions, if that is 0, then that would imply that x1 is equal to x2, because the 0 vector is the only 2k sparse vector in the null space of A. So that's the intuition behind compressive sensing. Now, in real testing, there will be some noise in the pools. In our RT-PCR tests, so there is no noise in the negative tests. So the zero valued pools are exactly 0, but the positive tests, they will have some noise in them. So for these noisy observations, you need there are slightly more uh, involved techniques which are known to us. So we can solve it using the so-called basis pursuit denoising, which is you do L1 norm minimization of X with this constraint that your observations must match close to at most epsilon value. And again, this can only be solved if my original vector x star, it was k sparse, and if my matrix A has the so-called restricted isometry property and of order 2k. So restricted isometry again means that this transformed vector Ax must have the same or close to the same length as the original vector x. So this delta 2k constant that you're seeing, that must be close to 0. And there is some guarantee regarding the solution that the solution that we have, it will be close to the original the unknown vector uh, in the L2 norm. But it depends on the amount of noise that you have. If you have more noise, so this epsilon is uh, an estimate of the amount of noise that you have. So if you have more noise, then this solution will be poorer. And if your delta 2k, this restricted isometry constant, if that is close to 0, then you'll have a good solution. If it is closer to 1, you'll have a worse solution. So we solve, instead of this constraint problem, we solve this penalized problem, which is the well-known lasso formulation. And furthermore, for our problem, this x vector, we know that it is non-zero. I mean, the viral loads cannot be negative. So we do what is called a non-negative lasso. So again, compressed sensing also takes order k log n pools, and uh, it also gives you a quantitative estimate of x. So that's the main benefit. And if we compare the two methods, compressed sensing and comp, then we see that compressed sensing can take advantage of the quantitative information for decoding. But it doesn't take advantage of the fact that these zero valued pools are exactly 0. It actually assumes some noise in these zero value pools as well. So it may happen that it gives you some false positives. So can we combine these methods? And that is our work, Tapestry. So we combine both comp and compressed sensing. So what we do is, first we look at all the negative tests. So we first apply comp, Right? So we look at the negative test, and whoever has taken part in the negative test, we eliminate those samples, and we eliminate that test as well. So because we know that those samples are zero, so we get this reduced matrix. And again, this reduced matrix, using that we can solve for this unknown vector x4, x5, x6. Uh, so that is the tapestry method. So it turns out that it's better than both comp and compressed sensing individually. So comp helps the compressed sensing step by eliminating the obvious negatives, which compressed sensing could not do. And also it speeds up the compressed sensing step by reducing the matrix. And in turn, what the compressed sensing does is that with comp, you may get many false positives. Uh, so the compressed sensing step eliminates some of those false positives, but it introduces a risk of some false negatives. And also the restricted isometry property that I talked about, that is preserved by comp. So the reduced matrix also has a restricted isometry property, which is why we can apply compressed sensing on the reduced matrix. And this also needs order k log n test. So we'll just talk a little bit about preservation of restricted isometry. So we are given that our original matrix A has the restricted isometry property, and now we do comp and we get our reduced matrix, right? And we want to prove that this reduced matrix also has the same property. So if you consider any sparse vector Z, whose number of dimensions is equal to the number of columns in the reduced matrix, and this is S sparse. So there are S entries which are non-zero, and you extend this vector Z so that the number of dimensions is equal to the number of columns of the original matrix, and you extend it using zeros. So the new vector Z tilde, that is also S sparse. And we know that because it's an S sparse vector and A has the RIP, hence this must be true. This is just the statement of the RIP. So now we know that really this is trivial to see that Z tilde and Z must have the same norm. And furthermore, the transformed vector A times Z tilde and A reduced times Z, they also have the same norm. Why is that the case? Because there are zeros here in Z tilde, which don't contribute to the matrix multiplication. And there are zeros here in A in the columns of A reduced. So here, if there was a 1 here, then since this is a negative test, then this column would have gotten eliminated. It would not have been in A reduced. So all we are left with are 0 entries here. So which is why this part is not contributing anything to the matrix multiplication. So which is why these two multiplications are the same. They have the same norm, and hence we can just substitute it and we get our property for the reduced matrix. So let's just look at some comparison between our tapestry method and comp and compressed sensing. So we see that, first of all, comp does not give any false negatives, and k here is the number of infected samples. So comp does not have any false negatives, so we haven't included that column. So it only has false positives. And our tapestry, which is this comp and then lasso, that has some false negatives. So you can see that if we use only the NN lasso, which is compressed sensing, then you get many false positives, and you also get some false negatives. But if you combine them, then then you get fewer false negatives than the NN lasso method. and you get fewer false positives than the comp method. So this is better than either of them. And this example is for a matrix which had 93 rows and 961 columns. So you get around more than 10 times improvement in testing. And again, we have some numbers for a 45 by 105 matrix. So this is only 2 times improvement. However, the number of infected samples that you can handle in relation to the number of samples that there are is larger. So you see some similar numbers here. And lastly, to compare with Dorfman testing, we see that for Dorfman testing, for 105 samples, in general, you need more than 45 tests. And for 961 samples as well, you need a lot more than 93 tests. So, however, Dorfman testing does not have any false positives or false negatives. So there is that trade-off. So finally, I would like to talk about how we chose our pooling matrix. So it should have these properties. It should be sparse, which will reduce our pooling effort and conserve reagents as well. It should be disjunct, and it should have the restricted isometry property. So we chose this matrix called Kertman triple system matrices. So I'll not talk about what they are, but really we proved a bunch of nice properties about this matrix, that they are two disjunct, they have restricted isometry, and furthermore, if an F fraction of the tests are positive, then it eliminates 1 minus F square fraction of the sample. So which is really nice. And also this matrix, you can serve part of this matrix as well while maintaining these properties. So that's also very nice about this matrix. So we had deployed this at Triple IT Hyderabad. This is the work of Professor Manoj Gopalkrishnan and CCMB. And so the outcome of this was that we could control the amount of COVID cases there. So yeah, so lastly, we have some other applications of pool testing. Now, unfortunately, I'm not a biologist, so I don't really know much about this, but I have added it here so that it can be useful to you. So there are many applications, and there is this book, Pooling Design and Non-Adaptive Group Testing, which gives a lot of applications of this. So yeah, and many other people were involved in this work. So thanks to all of them. Thank you, guys. That's my talk.
Thank you very much, Sabyasachi.
Yeah.
I don't see any questions posed by the audience in the chat box. If anybody has any questions, I would like to request them to please post those into the chat box. I do have a couple of questions myself. So
Yes.
Sabyasachi, I think so the novelty of tapestry pooling, as I understood, was you used a combination of on the first level you were using comp to isolate the negative test, and then you were using compressive sensing. In compressive sensing, you went for first lasso and then NN lasso. What was the improvement that you observed when you used NN lasso as opposed to just plain vanilla lasso? And the second part of the question, did you try using adaptive lasso or random lasso?
So no, so we only tried NN lasso. Other than NN lasso, we tried sparse Bayesian learning, as well as NN OMP, which is non-negative OMP. So actually, we didn't try pure lasso because we had this non-negativity constraint, so we used it. So actually, out of all of our algorithms, the best which was the sparse Bayesian learning, which gives the most balance between false positive and false negative.
Okay.
Yeah.
I, hmm.
Yeah. Have I
I
answered
do.
all the questions? Yeah.
Yes, yes, you did. I don't see any other question. Okay, there is one
Okay. There
question
is
by Michelle.
one in the chat.
So he's saying that it seems that you have to split a single individual sample into multiple portions to use these methods. How many portions do you use?
Yeah, so that's basically the pool size in our matrix. So in the 45 by 105 matrix, that's around it's 7. In the 93 by 961 matrix, it's much larger. It's 31.
Okay. There is one more question. So firstly, thank you so much to Alexis Casas, who has put a link to your paper in the chat box. Thanks so much, Alexis. Karthik has one question. If the contact map is partially or fully known, can it be used to constrain the problem better and
Yes.
obtain further?
Yeah, so that's true. And in fact, Ajit, so my advisor, Professor Ajit Rajware, he has some work on this, on contact tracing and using that. Uh, so it does give some improvement. If you want, I can share his work on this chat later or in the Slack.
Okay, okay, thanks. Then there are two more questions. One question by Brett Peterson. He's requesting you to give us a little bit of information about the noise level.
Yeah,
Okay, he
yeah.
believes it would be very high.
Yeah, yeah. So yeah, you're right. So the noise level is quite high. So in our this synthetic data, what we did was we used an exponential noise. So because so what we did was we assumed so RT-PCR process, it gives cycle threshold as the quantitative output. It doesn't give you viral load. So that we assume some noise in that cycle threshold, right? And if you can see that if that cycle threshold is off by 1, right? And because of doubling, because of RT-PCR amplification in every cycle, you can see that the viral load of the pool will be off by a large factor. Uh, so that is true. However, we found that thankfully it doesn't affect the support determination by that much, and most of the noise in this process is in samples which have very low viral load. In samples with high viral loads, thankfully we don't have as much noise.
Okay,
So yeah.
yeah. There's one more question, but before I pose that, could you please stop sharing your screen?
Okay.
Okay, so one last question we'll just take now. This is by Mahanas. Okay, the question is that we have an estimation or simulation to know up to what percentage of positive COVID cases in population or sampling is it possible to do such pooling, considering time and resources?
I didn't quite follow the question. Let me just check the chat. Yeah. Sorry, can you just repeat the question? I lost my chat window.
Sorry, yeah. So the question is up to what percentage of positive COVID cases in the population or sampling is it possible to do such pooling? I think so. Effectiveness of
Yeah.
pooling versus percentage of positive cases.
Yeah. So the results that I showed you, they are so the large matrix, the 93 by 961, it's valid till around 1% or 1.5%. The smaller matrix that works till, I mean, you can stretch it to around 10%. In theory, you can do up till around 30% or 33%, I think is the limit, which is the theoretical limit.
Okay. So thank you so much, Sabyasachi. We have overrun our scheduled time slot for the first talk by about 5 minutes. I'll quickly introduce our second speaker now. The second speaker is Maziya Ibrahim. Maziya has done her PhD in computational biology, and she's currently doing her postdoc at IIT Madras with Professor Karthik Raman, who is also attending this this workshop. All yours, Maziya.
Yeah, hello. I hope I'm audible. Yeah, so I'm Maziya. I'm going to present my work on microbial community analysis for metabolite production. My talk will be in two parts. So basically, I'm going to give an introduction on constraint-based modeling, what is genome scale metabolic models and flux balance analysis, and segue into our work on community analysis for metabolite production. So what are genome scale metabolic models? So the modeling begins when you have an genome sequence of an organism of interest. And the annotation of this genome will give us information on what genes and gene products, that is enzymes that are present, and what these enzymes can convert a product A to B. And this is done with the help of different knowledge bases such as KEGG. The mapping of this to reconstruct a network of all known reactions with shared metabolites is what is the genome scale metabolic model. And this is these models can also be compartmentalized into compartments such as the cytosol and extracellular space. And also even other these two compartments are the main ones seen in most models. You can also have compartments for the mitochondria, endoplasmic reticulum, etc. There are available tools for automated reconstruction of these models given a genome sequence. You have databases like Model Seed, which can do such an automated reconstruction, and you also have different knowledge other databases such as the big models, which store a lot of curated genome scale metabolic models. So why build such computational models? It's because it helps to organize all the metabolic information into a coherent whole. It's a formal representation of the metabolism of an organism. And with the on the basis of the coverage and quality of reconstruction, it helps in performing many simulations to understand the metabolism.
Maziya, sorry to interrupt you, but there is some background noise when you speak. Sometimes the voice comes very clearly. Otherwise, I think as you move the cursor, maybe at that time there is interference and noise.
Is it fairly better right now?
Yeah,
maybe hold the mic in your hand, it should be fine.
Yeah, okay. Yeah, so one of the important usefulness of genome scale metabolic models is identifying the essential genes and reactions which can help simulate different perturbations in the metabolic system. To understand how we use a genome scale metabolic models, we need to understand what is flux balance analysis, which basically forms the crux of many constraint-based modeling algorithms. And if the genome scale reconstruction is considered as a set of reactions, such as A giving B plus C, B plus 2C giving D, this can be mathematically represented as a stoichiometric matrix, where the rows are the metabolites and the columns are the reactions, and the coefficients of these metabolites denotes if it has been consumed. It's negative. If the metabolite is produced in the particular reaction, it's positive, and if it isn't taking part in the reaction, it's a zero. The reaction called biomass represents all the metabolites that are needed for the biomass or the growth of the organism. And we can also have the extracellular uptake components such as glucose and oxygen, which would be required for the growth of the cell. So at steady state, this system is represented as S dot V equal to 0, which is V is a vector of the fluxes of each reaction in the model. And so all of these equations are, it's an underdetermined system because the number of reactions is larger than the number of metabolites. And typically this is solved by using an objective function, which is given as Z equal to C transpose V, where C is a vector of weights for each reaction's contribution to the objective. And in practice, the objective function is usually the biomass of the cell. And so C would be a vector of zeros with 1 against the biomass reaction. So this is solved using linear programming methods, and the different constraints that are applied are the extracellular flux constraints, as well as capacity constraints, like based on the reversibility and irreversibility of the reactions. And solving this problem identifies an optimal solution, which in most cases is a maximization of biomass of the cell. So for any bio-based chemical production, what we can see is that genome scale metabolic models are very useful, as they are part of an integrated computational and experimental approach for production of bio-based products. And the simulations can help can be helpful in predicting strain engineering targets, which can be validated experimentally. So the targets can be of different kinds. So you can have bulk chemicals, fine chemicals, biofuels, polymers, and different substrates that can be used for production of these products. To select microbial strains, there's always preference over strains which can naturally overproduce a product versus strains which produce the product with low efficiency or which does not produce the product. Such microbial strains would require engineering for the desired metabolic phenotype. So while a lot of studies have been done with single microbes for production of these chemicals, recently there is co-cultures which are used because there's a lot of division of metabolic labor that can happen. There's effective conversion of substrate to product, and which can also result in reduction of the cost. So with this in mind, since this workshop is on machine learning applications, I just want to comment that there is a lot of integration of machine learning with flux balance analysis, both unsupervised as well as supervised. And there are a lot of papers out in the last few years on this. While the work that I'm presenting today is not classically machine learning, what we do is a more of a combinatorial evaluation, where if we are given a set of such genome scale metabolic models, we are trying to identify suitable two-species communities or co-cultures that can produce a given metabolite. So as case study, we have used lactic acid bacteria models for the production of lactate. We've taken models from the Lactobacillus, Leuconostoc, and Pediococcus genera. So this is a brief outline of the analysis. The first step we do is we do monoculture simulations, where we've taken the genome scale metabolic models from public repositories. We use 3 different nutrient conditions, which I'll elaborate in the next slide, and we do a flux balance analysis. In the second step is where we generate the communities. So we use algorithms such as SteadyCom to generate all possible communities from the given set of models. We again use 3 different nutrient conditions and FPA. And this will help us compare monoculture and co-culture growth rates. The third step, we do a community assessment. So we look at how much is the observed metabolite yield in the co-culture versus the expected. We can also look at interaction behaviors, mutualism or parasitism, depending on the growth rates. We also look at cross-fed metabolites. And in the fourth step, we do an in silico community optimization, where we use algorithms such as FSUF, which helps us predict suitable reaction knockouts, which when deleted will improve the flux of the product. So our data was involving 49 different models. We used carbon substrates, glucose and xylose, and we used 3 different nutrient conditions. Since we are not using any experimental constraints, we have used these 3 these 3 conditions. The minimal condition involves giving an uptake of -1 for glucose and xylose. The excess nutrient condition is where we give 30 for glucose and 10 for xylose. This is to sort of mimic the composition of glucose and xylose in lignocellulose hydrolysate, which is the common substrate used for production of lactic acid. And we also have a community-specific nutrient condition where we find the glucose and uptake, xylose uptake fluxes at half maximal growth rates for each model. We use ATP constraints as 0.36, and these simulations are performed in an anaerobic environment. Steadicom helps us generate 1,000 odd communities under these three conditions. So what we see is that the active pathways, that is the reactions which carry flux in all our simulations, belong to the central carbon metabolism, that is the EMP, the PPP, and the pentose phosphoketolase pathway. And this is expected since we are providing substrates of glucose and xylose, and our end products are mainly lactate, acetate, and ethanol. Based on the growth rates that we observe in monoculture and co-culture for these models, we identify different community interaction behaviors based on a significant change of 10%. So mutualism is when both the organisms have an increase of 10% in co-culture versus monoculture. Commensal when one organism is benefited while the other stays the same. Parasitism when one organism is benefited, the other is having a lesser growth rate. Competition is when both of them have lesser growth rates than monoculture. Amensalism when one organism has a lesser growth rate, and neutralism when both of them have no significant change. We pick up viable communities from those 1,000 odd communities. So viable communities are those which have at least a 0.01 growth rate. So when we compare the monoculture and co-culture growth rates, this heat map shows the organism on the y-axis when it is co-cultured with the organism on the x-axis. If there is an increase, it's colored blue. If there's a decrease in growth rate, it's red. And if there's no significant change, it's in gray. The white squares are non-viable pairs. And we did this for the three nutrient conditions. And we see that there are around 300 plus pairs which are viable. And parasitism seems to be favored in the community-specific and minimal nutrient conditions, and amensalism is favored in the excess nutrient condition. The algorithm Steadicom also provides us with abundances of each species in the co-culture. And when we plot the abundances of each model in the three different nutrient conditions, we can see that the abundances are varying depending upon the number of viable communities that is associated with each model. And there's also a difference in the abundance depending on the nutrient condition. So this kind of highlights the role of the nutrient constraints in driving the community behavior. We also did simulations where we provide a single substrate like glucose or xylose, and also when both glucose and xylose are provided. And what we see is that in xylose-only condition, there's a lot more of mutualistic pairs that is observed. And this indicates a sort of a rise of an emergent property in the community, since most of the models are not typically capable of growing on xylose alone. And we see more of competition arising in glucose excess conditions because all the models can grow on glucose. We also look at what metabolites could be cross-fed. So within each organism, you have the cytosolic and extracellular space. And in the community, in the two species community, you have a community space, community compartment U. So we define a cross-fed metabolite as one which is secreted into the community compartment by one organism and it's taken up by the other. And what we see is that the cross-fed metabolites are succinate, malate, formate, ethanol. And the fraction of metabolites which are cross-fed in cooperative communities are higher than those in communities which exhibited parasitic or competitive behavior. The other thing that we looked at is of the lactate yield, and we computed the expected yield based on the monoculture lactate yield in each model. We used the Kanye model proposed by Medlock et al. And so the expected metabolite yield is basically the sum of the product of the substrate uptake of this first species in co-culture times the yield of the metabolite in the first species in monoculture. And similarly with the second species. And the observed yield would be the total metabolite flux, which is observed in the co-culture, divided by the substrate uptake flux. And we picked up communities where there was at least a 10-fold increase in the observed yield compared to the expected yield, and we considered those pairs as candidate pairs for the production of that metabolite. What we also did was we considered one organism in the pair as a primary consumer and shut off the uptake for glucose and xylose by the second organism. So this sort of creating a dependence of one organism on the other, and communities which still remained viable where both these species were growing were deemed fit for further optimization strategies. What we observed was that our top ranking pairs based on the previous parameters were those where both the organisms could ferment glucose, and not really were the ones where both xylose and glucose are utilized. And the top pairs were comprised of the Lactobacillus plantarum species. We also computed metabolic distances between these models in the top pairs, and these are basically the differences in the reactions between the models. And we find that the Chakar distance is greater than 0.7, which shows that they exhibit distinct metabolic capabilities. And these top ranking communities had displayed mostly commensal or mutualistic behavior. So we can say that metabolic complementarity and compatibility between the organisms is necessary for the stability of a community. When we looked at what drives the flux, what drives the high lactate yield in certain communities versus the others, when we look at the comparison in the fluxes of the reactions, we see that there is a 5-fold increase in certain reactions which are basically part of the carbohydrate metabolism, such as glyceraldehyde 3-phosphate dehydrogenase, triphosphate isomerase, fructose bisphosphate aldolase, and phosphoenol pyruvate carboxylase. Finally, we use the FSCF algorithm, which helps us predict the reaction knockouts that can be deleted to improve the lactate yield. And FSCF is traditionally used for single models, and here we've extended it to use for microbial communities. And we see that it predicts reactions such as acetate kinase, pyruvate formate lyase, fumarate reductase, etc., which are basically these reactions have also been seen to increase lactate yield in other species, experimentally validated in other organisms such as E. coli. And we see that we even in communities, it's the similar reaction knockouts that is observed, which will impede the flux towards acetate formate succinate and increase the flux towards the biosynthesis of lactate. So to summarize, we have put together a framework where we're looking at community growth as the objective, and the community is tested for growth on 2 carbon sources. We are able to screen viable communities based on their growth and lactate yield. And we are using FSCUF to predict reaction knockouts to improve the product flux. And these gene knockouts, gene or reaction knockouts that are predicted which are identified in monoculture, can also be extended to microbial communities. And these knockouts could either be from one or both the organisms in the co-culture. Yep, that's it. I'd like to acknowledge my Professor Karthik Raman and the members of the Computational Systems Biology Lab and Center for Integrative Biology and Systems Medicine at IWBDA. I'd also like to thank Professor Vishwesh and IWBDA to give me an opportunity to present my work. And I'll take any questions.
Thank you so much, Maziya. I don't see any questions in the chat box, but I do have a question of my own. Could you please go to slide number 22?
Yeah.
Okay, so in FSCUF, you're using reaction knockouts. I assume you are doing the single reaction knockouts. Have you tried knocking off pairs of reactions or more than one reaction at a time?
Yeah. These are either single or two. So it could be like acetate kinase from one of the models, and fumarate reductase from the other model. So yeah, I have tried for two. And single and double, not more than that.
And what is the running time of the algorithm on this data set for those algorithms?
So it depends on the input number of models. So our models that we've used is like 49, and it isn't much. So you can complete it like within a day. You can complete everything within a day. But the limitation would be like, yeah, if you have a lot of models, like in the hundreds, then it will take some time.
And you're using normal computer to run the algorithms, and it takes approximately 24 hours, right?
No, to do everything. To just do Steadicom, it'll probably just take you 2 hours maybe.
Okay. All right then. So thank you so much again. I think we'll move on to the next talk. Could you please stop sharing the screen now? The next talk will be given by Leon Faulon. He has background both in experimental as well as in computational biology. He's currently a first-year PhD student in the laboratory of Professor Jean-Loup Faulon, and he's currently working on applying machine learning for bioengineering with examples from the circuits that are being built and developed in the laboratory of Professor Jean-Loup Faulon. So all yours, Leon.
Thank you. I will share my screen. Okay. Okay, can you see the screen?
Yes.
Okay, so first of all, thank you for inviting me and the group of Jean-Loup to this conference. So the talk will be a general overview of how do we apply machine learning for bioengineering in our group for the past few years. So first of all, there are three main categories of machine learning that we applied to biological objects in our lab. So first, the first one is the most traditional classical way of machine learning, which is supervised or unsupervised learning. So in this category, we basically acquire large amounts of data from a biological database, and we apply classical algorithms of learning to eventually predict new experimental work that will be useful for discovery and annotation. But most better suited to our work in the lab is the active and reinforcement learning, where you basically acquire the training set on the fly, on the go. So it's particularly suited to synthetic biology and systems biology because the ground truth that you are learning is actually the experiments, and since it's it's taking a long time to basically perform the experiment itself, it's very useful to use this kind of learning because you can acquire the set on the go. And finally, the third type of learning that I will present is very different. It's basically introducing components into biological systems that perform the computation itself. So it's in vivo or in vitro learning and computation. So it's a very different type, which has been notably the first group that has done this kind of work was with DNA strand displacement. And for us in this talk, it would be about metabolism and enzymatic transformations that actually perform the computing. So first of all, I will describe a bit about the supervised learning. So here I show an example of a training set of annotated sequences annotated with their EC numbers. So we have in this training set about 4 million sequences, And basically, the first thing to do is to encode the sequence so as it is suitable for a machine learning model. So here I show a sliding window that basically produces string spectrum. But you can also encode the sequence with the PFAM domains or with the one-hot encoding. There are a lot of different techniques that exist to encode the sequence. So the training is, we are using different kinds of models, but the most famous one is the neural network, which basically each node is doing weighted sum from all the previous nodes, all the connected nodes, and the final node is actually very important as a very important activation function, where for classification, in this case, we need a 0-1 answer, so we use a sigmoid function. So the test set is an independent set of data, which is very important to separate it from the training set to avoid overfitting. So here is just a result from the prediction of the EC number from the sequence itself. And we do the validation, so using this accuracy formula, so we can reach very good results. Another kind of learning can be it can be used without the EC number information, because basically EC numbers have pitfalls, and we can use directly the reaction information and not just the EC number. So for that, we need also, along with the sequence information, we also need the reaction that is catalyzed by the reaction. So this can be stored in various databases, such as Brenda. And we need also to encode the reaction. So for that, we can use the ECFP, which is the extended connectivity fingerprint. So this basically encodes the molecular signature of the reaction into bit vectors that can be fed to the neural network model. And to actually simulate the interaction between the sequence and the reaction, we use the tensor product. So the tensor product is a basic operation to actually merge the two vectors. And we have published a paper with Gaussian processes, so not neural networks, but Gaussian processes that performs well to automatically screen for the interactions between the sequence and the reaction. So you can see here the results on the left. The GP is for Gaussian processes, and it outperforms the support vector machine. So how do we apply then? How do we apply this Gaussian process model? Was to basically screen for new metabolites in E. coli. So basically, the underground metabolism is the metabolism performed by promiscuous sequences. So where unexpected metabolites can be produced by enzymes. And here we discovered 3 enzymes according to the model that would produce N-acetyl-L-leucine. And basically the experimental validation here showed that we do find N-acetyl-leucine in excess when we overexpress these 3 enzymes. So it's actually, the model is not only performing well in silico, but also it was used so to discover new capacities of enzymes. So now I'll go to the second type of learning. So this is called active and reinforcement learning. So in a nutshell, it's basically algorithms where you train the model iteratively and acquire a growing training set as you perform the experiments iteratively. So the active learning is fairly, it's been known from a long time in chemistry. For example, it has been used to discover kinase inhibitors in drug developments. And reinforcement learning is more sophisticated. It's basically using more fine tuning of the model, because we use cumulative rewards and basically tree search in the state-of-the-art models, such as the AlphaGo the AlphaGo model developed by Google. And we also published reinforcement learning for a bioretrosynthesis project published in 2020 by Mathilde Corr. So I will go in more detail into the active learning. I will not have time to show about to speak about reinforcement learning. So active learning in Jean-Loup's lab has been used mainly for cell-free productivity. So cell-free is basically transcription translation systems with basically E. coli cell extract that you mix with a few components, so NTPs, salt cofactors, amino acids, and DNA for expressing particular enzymes. Basically, by mixing all of these, the reactions, the biochemical reactions, can happen without living cells. So it's more stable and robust, and it has a lot of potential for bioproduction. Basically, we selected 11 components with different concentrations, starting with this reference concentration that is from the literature. But if you change these concentrations, so you can divide by 2, 3, or 10, you obtain a combinatorial space that is huge, that is more than 4 million compositions. So obviously you cannot screen all these compositions with experimental work, and the goal of active learning is actually to guide the exploration of all these compositions with the machine learning procedure. So 2 questions emerge from the active learning on these compositions. Is, can we find a better set of concentrations with our model? And also, can we reduce the price of the reaction? So that basically means reduce the concentration of the components, especially for the expensive components. So now the actual process will be described. So the first step is to set up the initial batch, sampling the space of the compositions. So you can use the literature, or do a random search, or use fractional design, which is a classical method in designs of experiments. So this would be the first training set for your model. You will measure the yield, so actually do the experimental work and measure the yield with fluorescence, since we use, in our case, the expression of a fluorescent protein. Then you will fit the neural network to the measured yield. You will use then the model to predict the yield for every other compositions that have been tested. Then the next batch of compositions to be experimentally measured will be based on the upper confidence bound formula, which is basically a trade-off between the exploitation or the exploration. So the exploitation means that your next batch will be maximizing the mean, so the actual yield, so maximizing the productivity of the system. And the exploration means that you will actually maximize the variance between the different models. Sorry, a very important thing is that you have a set of 25 models that enables you to have a mean and a variance on your predictions. And so the uncertain prediction would have a high variance, and the exploration term is giving to your next batch of experiment the points where the variance is the highest between the models. And this is useful because you will explore the parts of the space that are the least certain, and you will improve the predictability and the accuracy of the model. And so then you can repeat this process a number of times until you reach the satisfactory results, or you converge to a plateau that you cannot go higher. So these are the results. So you can see that in about 3,000 measurements out of the 4 million possible compositions. So sorry, 3,000 measurements in triplicate. So it was actually only 1,000 different compositions. We could increase the yield times 34. So it's a very substantial increase of the yield from the literature composition to the top one that we found after just 7 iterations. And so you can see here the evolution of the concentrations. So the more red, the lower is the concentration, and the increase in yield through the different compositions that have been tested. So you can see also on this figure that we not only increase the productivity of the self-read system, but also it's the accuracy of the model. The R square is higher than 0.9 after the 7th iteration. And also you can see on this figure that we can extract some real insight of what components are critical into the reaction with this mutual information score, showing that the tRNA, the NAD, and folinic acid are not critical into the reaction, and so that we can lower the concentration in order to reduce the price. So now I will go to the third part and last part of the talk. So this is a very different type of learning. This is actually a learning that is used to compute in vivo. The actual computing will be done in vivo. So the first important thing to know is that biomarkers can be used to predict the severity or the outcome of a lot of different diseases. For example, here you can see for prostate cancer that you can measure this metabolite, and it gives you some pretty reasonable performance for the screening of the prostate cancer for the diagnostic. But if you combine different biomarkers, you increase these results. So using a classical perceptron, so which is the classical architecture in deep learning, the most basic unit of deep learning, sorry, you can actually predict the diagnostic based on a combination of different biomarkers concentrations. So you can perform the diagnostic by quantifying this panel of biomarkers using metabolomics. So then you will feed the measurements X, so Xi, and so you will be able, with the output of this function, be able to diagnostic the disease. But for that, as I said, you need metabolomics. And the question we wanted to answer was if it was possible to avoid this metabolomic measurement and engineer basically a biological system that is doing the same computation as this trained perceptron, but directly with the biomarkers and directly in vivo or in vitro. And so the first step to do that is to transform the perceptron into an actual biological system. So you need two things actually to be able to reproduce this behavior into a biological system. You need an actuate weighted sum and also an activation function. So for the activation function, which is in our case a sigmoid for diagnostic, we are good because basically the reporter gene, GFP, in our case, will be transcribed by a transcription factor that has a sigmoid behavior according to its fold change. So basically the transcription factor will induce a sigmoid behavior to the fluorescence. So for that, we can use this GFP directly. And for the enzymatic transformations that perform the weighted sum, in theory of the Michaelis-Menten theory, when the substrate is in very lesser concentration than the enzymes, we can say that the weights correspond to actually this term, the kinetics constant times the enzyme concentration. So as you know, the kinetics constant is very hard to change. It needs enzyme engineering. So we use simply the enzyme concentration as the weight in our system. So here is the actual system. You can see this scheme here describing the system and the results of the measurements and the model, because we needed to model the behavior of these enzymatic transformations in order to translate the weights of the perceptron into the levels of enzymes that correspond. So you can see that for a given weight, so actually the weight, I said that the weight was the enzyme level, but it's actually, since we are in the cell-free system, it's actually the amount of coding DNA for this enzyme. And so here you can see that for 3 nanomolar of the enzyme DNA, we obtain a nice range of fluorescence. So the fluorescence is given with the color, and yeah, for a given weight, you can have a nice range, and by changing the weight, you can the response of the system would be changing, and we can actually use that to engineer our biological system. You have to be careful because of resource competition in cell-free systems, you cannot just simply increase the DNA to increase the performance of the enzyme, because yeah, there will be a saturation point when the GFP will be not produced, and it will be only the hyperic acid degrading enzyme that will be produced. So we basically, in the paper, so it was published by Amir Pandi and al. In Nature Communications in 2019. And in this paper, we implemented a binary classifier that was implemented really in vitro, and that works. So I will show you a bit how it works. So the first thing is to target a behavior. So this is predefined. This is completely arbitrary. We want the system to have a negative response for this case, so when you have only hyperate or cocaine, and we want to have a positive response for all these other cases. And so by negative response, it means less than the threshold, 0.5, and positive is more than 0.5. So you have this targeted behavior, and this targeted behavior can be translated into weights, into actually be the bias and a set of weights. This set of weights with the models I showed before can be then transcribed into the enzyme concentrations corresponding to each weight. So coding DNA for the enzyme. And then you can actually directly measure the fluorescence to see if it fits the targeted behavior. And it's the case. Here you can see that the RFU is above the threshold for every positive example, and it's less than the threshold for all the negatives. So we actually engineered a working perceptron into a biological system. So that's it for me. Thank you for listening, and I will be happy to answer your questions.
Thank you so much, Leon. I have just one quick question. So you mentioned active learning in the context of cell-free optimization. I think you are using random sampling. Is that correct? On the slide, I think I saw random sampling somewhere.
Sorry, yeah, for the first initial training set,
yes,
you can use either, you can just generate compositions randomly, since it's the initial set, and it matters less than the next batches, because the next batches will have the help of the model to guide you. But the first round can be either done with literature references. So if you need, if you have well-performing compositions that you can find in the literature, that's good. Or you can use a fractional design, or you can use randomly randomly generated compositions. Yeah.
And beyond that stage, then do you use pooling all the same? So for example, typically in active learning, we can use entropy-based pooling, or we can use LC-based pooling. So which strategy did you use?
You mean pooling of what?
So pooling of the samples. So in each iteration of active learning, you would have to group them.
Mm-hmm.
So do you implement that, or?
In this paper, we didn't pool the samples. We just use them independently. I mean, just yes, the triplicates were pulled together, but except from that, I think we didn't pool. No, I'm sorry. Yeah, I don't think we pooled any sample.
Okay, and if I understand correctly, so this code is available in Python, and you have released it on GitHub, correct?
Yes, we do have this. Yeah, on the GitHub from the BioRetroSense team. Yeah.
Okay, thanks. I'm now taking a look at the chat box to see if anybody has any questions. No, I don't see any questions. So thank you so much, Leon. It was a really nice presentation, a lot of information. So we now come to the final talk of today's session. This would be a talk that is given jointly by Pablo Carbonell and Jonathan Delechia. Pablo is a senior reader in computational biology at Polytechnic University of Valencia. He was formerly a senior staff scientist at SynBio Center at Manchester Institute of Biotechnology. His research interests are in automated design for metabolic engineering and synthetic biology. The second part of the talk would be, or the other part of the talk would be given by Jonathan Delechia, who is a postdoctoral researcher in Pablo's lab in Valencia. Jonathan holds a PhD in synthetic biology from Newcastle. So Pablo and Jonathan, I request you to please start your talk.
Thank you. Thank you very much for the presentation. So I'm going now to share my screen. I hope that everybody can see it. Is that fine?
Yes, it looks good.
Yes, it looks good.
Yes. Okay, thank you very much. So I'm going to talk today about a pipeline that we have implemented that performs the DBTL cycle, the design, build, test, and learn cycle in an automated way. So the outline of my talk is, first I will give you some ideas about biomanufacturing and digital twins. Then I will focus on efficient experimental design, and we'll have some discussion on optimal design and machine learning. And then I will pass it on to Jonathan, who is going to give you a hands-on example. So let me first start by talking about the smart biomanufacturing revolution. As we have seen in many of the talks in this workshop, so nowadays labs are using a lot of automation, including robotic platforms, and that generates huge amounts of data that then we can use in order to train algorithms for automated learning, which will then be used to build models, predictive models that can guide the in silico design in a way that will actually determine the experimental planning in the biomanufacturing. So this is something that more and more we can see in labs and biofoundries. And what you can see here is the way that we implemented this DBTL automated platform at SynBioChem at the University of Manchester, where basically what we performed was to identify the steps that you have at the design, build, test, and learn cycle typically, and then try to automate each task, starting from pathway selection, enzyme selection, DNA parts, design of experiments, assembly, then go to the build, DNA synthesis, part preparation, assembly, DNA purification, and sequence verification. And if everything goes well, then we go into the analytical part, the test, chassis transformation, culture growth, product extraction, mass spec screening, and data analysis and processing, which then is fed back into the learn step, where perform machine learning and some other statistical analysis in order to redesign the rules for the design step. So this is something that since we can perform that in a quite automated way, and intercommunication between the different tasks. What we can also implement is a virtual lab that actually reproduces every step or simulates every step like a digital twin of a biomanufacturing process. In that way, so you can try different scenarios and then try to optimize the process as much as possible, because if a biomanufacturing DBTL cycle is something that is expensive, if you can in silico perform it in multiple ways and thousands of different ways and simulate the outcomes. So that allows us to actually consider every factor that can affect the final performance of the process, not only to optimize what is the final process or the downstream process or the media and the culture, but also the bottom-up steps like pathway selection, enzyme host intervention, and parts regulation. So having more like a holistic approach in order to optimize the full process. Then, in order to do that, we apply strategies for biomanufacturing optimization. Typically, initially you will apply screening, and depending on the throughput of your facilities, either if you have low throughput, you can apply optimal design of experiments, or if you have high throughput, then you can apply machine learning if you have enough data in order to train the models. In a second step, once you have already identified and narrowed down the factors and the operating point, then you can go into optimization. Either by looking for linear trends, by applying factorial designs, or even looking for higher order interactions through response surface methodologies. Okay, so let me give you one example of applying machine learning to this type of processes. So here we try to predict either translational regulation or transcriptional regulation. In the case of translational regulation, so we want to predict the effect of the RBS, the ribosome binding site, for each gene in the expression of the enzymes that are resulting in this pathway. So what we did was to sequence an initial library that was generated in order to try to maximize the diversity, and then look for the actual nucleotides that change in this library in order to train a neural network. So the approach was, as it was already discussed in the previous talk, by using one-hot encoding. And you can see here, this was for the validation of the machine learning model, that all of these constructs that were not actually in the initial set actually were predicted with a nice accuracy. So I think we have an R-square that shows that the approach can work, okay? Of course, as you add more and more genes in these predictions, then things start to deteriorate, and you need to probably add more and more data. So the same goes for transcriptional regulation, and in this case, what we did was to predict promoter strength, and also to compare the promoter strengths that were predicted for constitutive and inducible promoters. So that's one approach that we can apply, and the other one is about using DOE. But what we want to do is to apply an efficient way of using the design of experiments. The DOE actually comes from looking at a trade-off between the cost of resources involved with your experiments and the precision that you want to achieve in model estimation. So how can we maximize the efficiency of these designs? And I will go to show you this for this example. This is a 4-gene pathway that produces a flavonoid, pinosembrin, where we apply the DBTL cycle in order to try to maximize the production of the flavonoid. So the first thing to consider was which were the factors that we were going to use in our combinatorial library. We selected the plasmids, promoters, and the gene rearrangement as the different factors that led to a combinatorial space of around 5,000 combinations. And we wanted to narrow down it as much as possible. So in order to do that, first, we encode the different factors using, again, one hot encoding as before, so that you have 1 or 0 depending on the presence or absence of each level in the factors. But in the case of the gene rearrangement, it's a little bit more involved, but what you can do is, for instance, to use mutual orthogonal Latin squares as a way of exploring as much as possible the design space of the rearrangements, but also having a good balance with the size of the full set of combinations that you are trying to explore. With that, then the algorithm that we are using for designing the experiment is based on the optimal design. That what it tries to do is to minimize the variance of the estimates of the coefficients, to minimize the error of the model that you are estimating from the experiment, which is the same as trying to maximize the information that is coming from the experiments. This is performed by minimizing metrics. There are several algorithms that are available, and more information will imply better estimation of the model, which in turn implies less prediction error. So everything here is related. So we tried just with a library of 16 members. So it's a very small library compared with the full set. And then there we look at the one that we're producing, the higher titers, the highest ones, and then look at the factors that were having more impact on them. So this is relatively easy to analyze in this case, and we saw that the backbone had a strong impact, followed by promoters in front of CHI, gene, and so on. This allows us to generate new design rules, okay, like select high copy number, backbone, keep CHI at the beginning of the construct, etc. And using them all together, so you can build a new library that has learned the rules from the previous iteration. And that was this 24 library that here we use this factorial design for the remaining factors. And finally, with this, we obtain a 350-fold increase once everything was optimized. So you can see here that just starting with a 16 library in the 24 library of the second round, we already see a good trend. Obviously, because it's so small, we cannot see a high precision, but it's already, we see that it's already going in the right direction, and you can see that on the fold increase. So as I said at the beginning of my talk, you can consider a virtual lab where everything is performed the same way, but through simulations, and then try many different scenarios, like different number of copy numbers, promoters, gene variants, number of steps, then generate all of these libraries by applying optimal design of experiments. And then we compare the simulated results in terms of efficiency, in terms of how much we can learn from these experiments. And you see here where you apply optimal design, that it appears a Pareto front that can be used in order to decide the practical size of the library that you want to implement, depending on the efficiency that you are expecting to obtain. So this is almost like a practical rule that can be used in the lab, just based on these simulations. And now with that, I pass it on now to Jonathan, who is going to present this hands-on example. So all is yours now, Jonathan. I'm going to stop my sharing.
Yeah. Okay. Hello, everyone.
Jonathan, I'll just step in quickly. So we are now at the 90-minute mark. So we are now in the grace period. It would be great, if you can please finish your talk within the next 10 minutes.
Yes, I will try. Yes, thank you.
Thank you.
Thank you, Pablo. Thank you for everyone. And yes, I'm here to just to give you a really short presentation of some hands-on examples of some of the steps that necessary to carry out a bioproduction project using synthetic biology tools and all the nice work that Pablo presented before. So for any given bioproduction project, one of the main questions would be pathway selection. So let's start from that. And so the idea of pathway selection is to choose the best metabolic route that fits best your host strain, your host organism, and producing the highest amount of metabolite. In this case, we are going to follow the pinosembrin example Pablo was describing before. And yeah, this task is not an easy task. It's complicated and there are many variables. And for that reason, some tools have been described, and precisely we are going to describe, to talk about a little bit about retropath and retro rules as well, which basically select and rank metabolic pathways towards, as I said, your chosen metabolite, ranking them through several features and characteristics like the theoretical production, the effectiveness of the enzyme, and the theoretical as well toxicity that the pathway could have inside your host cell. So once you have using any tool, in particular the ones I said before, to choose the best theoretical pathway, you need to start looking into the enzymes of that pathway. So maybe there's the route is 2, 3, 4 steps long. So you need to look into each of those steps and choose the best enzyme possible, looking at not easy to describe characteristics like reaction similarity, kinetics, taxonomical distance as well, from the original enzyme to your host cell, and also as well some other biochemical characteristics. So now I will try to do a small demo of cell enzyme. Cell enzyme is basically is a tool that selects as well enzymes and ranks them considering all these parameters I said before. So this is the first reaction in the pinosembrin pathway. So you just select, this is KEGG, so you can select many databases in cell enzyme. So I'm just using this one because I like it. So you select the identifier and you go to the landing page of cell enzyme and you select here KEGG or any other database that you want. You paste the identifier and it basically will identify the reaction you are trying to look into. And yeah, you select your host cell here and the number of hits that you want. Let's put just 10 to make it faster. And if you want or you don't want multiple sequence alignment as well. So it could also give that parameter as an output. You click submit and it's going to generate, as I said, at least of 10 candidates, enzyme candidates coming from different organisms, as you see here, that perform this reaction or a similar reaction that you can see here. So this is your query reaction, and this is this is your input reaction. This is the query reaction, and also the biochemical parameters, as I said, and the taxonomical distance. So it's a nice tool to bear in mind when you're trying to do this kind of projects. And of course, this is something that you have to go and try out in the lab. So the tools, both the RetroPath and Selenium, are suggesting which are the theoretical best candidates, but it's something that you always need to check in the lab. So both tools have you can work with them through command line, through Python libraries. Selenium, as I showed before, has a nice web application, and both tools, together with many others, are available in Galaxy SymBioCAD, which is basically a nice compilation and in the cloud, so you can work with them, plug them together. The input or the output of one of the tools is the input of the next one, and it's a nice applications published recently. So the next step, as I said, would be to go in the lab and try. Many of you have heard before, maybe even during these sessions, about SymBioHub and other repositories of DNA parts, where you can try and search for promoters, regulators, terminators, etc. Or you can even publish and share your constructs. So that will be the next step after pathway selection and enzyme selection. So up to here is, let's say, work done. And now I just want to show you a little bit more about where our group is going for, and where our group is going to in the next months or years. So we want to focus right now in biosensors, because we think they add a nice extra layer of complexity and potential features, nice features to have in any bioproduction project. So the first one is the possibility to do quick screenings instead of the production. So when you couple the production to the fluorescence of or the output of the reporter of the biosensor, you can have a quick screening process that tells you, okay, from these 100 colonies, which are the ones that are performing best. Of course, always you need to check in more analytical equipment like HPLC or mass spec. And another possibility with biosensors is the so-called dynamical regulation, which basically means that apart from producing biologically your compound, you are also making the strain more responsive. So you couple the biosensor with the production itself, and that means that you can control and make the cells more adaptable to changes or to some perturbances in the media or the environment. But however, the problem is that there are very few biosensors described for any given metabolite. And for that reason, that's what we are working on at the moment. And this is just a simple decision tree that we are using at the moment. Is if your compound has any biosensor described, then of course use it as a starting point, and you can play around with the regulatory parts upstream and downstream of the transcription factor. And then if there is no biosensor described, you can see if maybe in any plant or any animal or something like that, a transcription factor has been described to be triggered by your metabolite, and then use that to build your own prototype biosensor. And if that is not the case either, well, you can always try and look for biosensors or transcription factors in this case that let's say react or are triggered by compounds similar or closely related to your target metabolite. And finally, as a take-home message of this whole talk, I would like you to to consider that biomanufacturing, as we have seen in this talk and in previous sessions, is making good progress, is accomplishing and solving solutions of the bioeconomy, that these solutions are hugely due to DOE and machine learning collaboration and support, and that BioCAD tools and automated design are allowing researchers to build prototypes and to quickly identify issues early on in the process, allowing them to solve these issues before it's too late, let's say. And finally, just quick thanks and acknowledge all the collaborators of the group through the years. And yeah, we are happy to take any questions. Thank you.
Thank you so much, Jonathan and Pablo. I noticed that in the chat box, Pablo has kindly put in the link to cell enzyme, and he has also put in the link to a relevant paper as well. So because we are already almost 10 minutes over the time limit, so I won't pose any question from my own side. In the chat box, nobody else has posed any questions. So maybe it would be a time to wrap up this session here. I would like to express my sincere thanks to everybody who presented really nice presentations in this session today, and also for the audience. Okay. Thanks. Bye-bye.